morning. I'm Devasena. In this lecture, we are going to discuss about biosignal measurements. In detail, we are going to discuss about resting potentials and action potentials that are present in the cell. Biosignal measurements. Biologic systems frequently have electric activity associated with them. This activity can be a constant DC electric field, a constant flux of charge carrying particles, or a current, or a time varying electric field, or current associated with some time dependent biologic or biochemical phenomenon. Origin of biopotentials. Bioelectric phenomenon is of immense importance to biochemical engineers because these potentials are routinely recorded in modern clinical practice. Cells transport ions across their membrane, leading to ion concentration differences and therefore charge differences, hence generating voltage. Most cell groups in the tissues of the human body do not produce electric voltages synchronously, but more or less randomly. Thus, most tissues have a resultant voltage of zero as the various random voltages cancel out. When many cells produce voltages synchronously, the resultant voltage is high enough to be measurable. Example: EMG muscle fiber contraction. Most cells of the fiber perform the same electric activity synchronously and a measurable electric voltage appears. Here is this representation of EMG. Many organs in the human body, such as the heart, brain, muscles, and eyes, manifest their function through electric activity. The heart, for example, produces a signal called the electrocardiogram or ECG. The brain produces a signal called an electroencephalogram or EEG. The activity of muscles such as contraction and relaxation produces an electromyogram or EMG. Eye movement results in a signal called an electrooculogram or EOG, and retina within the eyes produce the electroretinogram or ERG. And the electric signals produced in the stomach is also called as electrogastogram. Measurements of these and other electric signals from the body can provide vital clues as to normal or pathological functions of the organs. For example, abnormal heartbeats or arrhythmias can be readily diagnosed from an ECG. Neurologists interpret EEG signals to identify epileptic seizure events. The origins of these biopotentials can be traced to the electrical activity at the cellular level. The electric potential across the cell membrane is the result of different ionic concentrations that exist inside and outside the cell. The electrochemical concentration gradient across a semi-permeable membrane results in the nernst potential. Figure shows the structure of the cell. as seen with the biological microscope. Each cell consists of a centrally located nucleus surrounded by the cytoplasm. The nucleus is separated from the cytoplasm by a nuclear membrane, and the cytoplasm is separated from the surrounding fluids by a cell membrane. The different substances that make up the cell are collectively called protoplasm, which is mainly composed of water, electrolytes, proteins, lipids, and carbohydrates. Water is the principal fluid medium of the cell, and its concentration is in between 70 and 85%. Water serves as a solvent for various chemicals to produce chemical reactions. The electrolytes present in the cell are potassium, magnesium, phosphate, sulfate, bicarbonate, and small quantities of sodium, calcium, and chloride. The electrolytes provide inorganic chemicals for the cellular reactions. Further, electrolytes at the cell membrane allow transmission of electrochemical impulses in nerve and muscle fibers, and the intracellular electrolytes demonstrate the activity of different enzymatically catalyzed reactions that are necessary for cellular metabolism. Proteins, which constitute 10 to 20% of the cell mass, are divided into structural proteins and globular proteins. Cell membrane. Here is a representation of cell. The cell membrane, also called the plasma membrane, is found in all cells and separates the interior of the cell from the outside environment. The cell membrane consists of a lipid bilayer that is semi-permeable. The cell membrane regulates the transport of materials entering and exiting the cell. Here is a representation of movement of the ions. The cell membrane separates high concentrations of potassium ion and low concentrations of sodium ions, along with other ions such as calcium in less significant proportions inside a cell and just the opposite outside a cell. Resting potential. The difference in ionic concentration across the cell membrane produces the resting potential. Here is a representation. Action potential. Some of the cells in the body are excitable and produce what is called an action potential, which results from emissions of these and other electric signals from the body can provide vital clues as to normal or pathological functions of the organs. We are going to see sinoatrial node excitation in heart. That is the action potential generated in the heart we are going to discuss. For example, abnormal heartbeats or arrhythmias can be readily diagnosed from an ECG. The ECG waveform is represented here. That is the PQRST waveform. Now we are going to see excitation of neuron action potential. That is action potential generated in the brain we are going to see. Here is a representation of excitation of neuron. Neurologists interpret EEG signals to identify epileptic seizure events. Simulation of muscle fibers, that is EMG electromyogram, that is action potential generated in the muscles we are going to see. EMG signals can be helpful in assessing muscle functions as well as neuromuscular disorders. Here is a representation of EMG signal. Factors influencing the flow of ions across the cell membrane are diffusion gradients, inwardly directed electric field, that is inside negative, outside positive, membrane structure, that is the availability of pores, potassium ions, sodium ions, and permeability of membrane to different ions. Active transport of ions across membrane against established electrochemical gradients. When appropriately stimulated, they generate an action potential, that is flow of ions across the cell membrane and generation of a propagating wave of depolarization along the membrane. Cell transport representation is shown here. The fluid which lies outside the cell membranes is called the extracellular fluid, and the fluid which lies inside the cell membrane is called the intracellular fluid. It is seen that the extracellular fluid contains large quantities of sodium and a small amount of potassium. But in the intracellular fluid, the concentration of potassium ions is more than the concentration of sodium ions. Similarly, the concentrations of phosphates and proteins are more in the extracellular fluid. Meanwhile, the concentration of chlorides is more in the extracellular fluid. To understand the origin of biopotentials, we need to focus on bioelectric phenomena at the cellular level, volume conductor fields of simple bioelectric sources, volume conductor fields of complex bioelectric sources, volume conductor fields as a necessary link between cellular activity and gross externally recorded biological signals. Resting state representation. Steady electrical potential of difference between internal and external environments represents the resting state. The value of the resting state is typically between -70 to -90 millivolts relative to the external medium. Active state representation. The electrical response to adequate stimulation occurs. It consists of all or none action potential after the cell threshold potential has been reached. All or none law. The all or none law is the principle that the strength by which a nerve or muscle fiber responds to a stimulus is independent of the strength of the stimulus. If the stimulus exceeds the threshold potential, the nerve or muscle fiber will give a complete response. Otherwise, there is no response. Thank you very much for listening this lecture. If you like this lecture means, kindly share this video to your friends. For further updates, kindly subscribe my channel. Thank you.